Today we've got Arnau Sebe Pedros, who's going to talk about single-cell atlasing across diversity. And then we have a session on symbiosis, and then a session on speciation genomics. So all processes which we're now managing to approach from the level of deep understanding with complete genomes as the base data. So Arnau, who's going to give the first keynote lecture, is a group leader at the CRG in Barcelona. He's also an associate faculty here at Sanger. He leads the Biodiversity Cell Atlas, which is a global initiative to promote cell atlasing across all of life and not just in model organisms. And I'm really looking forward to the talk.
Yeah,
thank you, Arnau.
Thank you very much. Yeah, so I'm very happy to be here today. Let's see if I will, yeah, it's only 6 minutes. I want to tell you a bit about the work we do in the lab, and I also hope to convince you how or why some of the work that you guys do in producing super high quality genomes are essential for what we do, which is essentially try to understand how these genomes, how the regulation of these genomes and the diversity of cell types that they encode evolve over time. So kind of the unit of study, what we focus in the lab, I always like to say that this is the phenotype, the molecular phenotype that we study in the lab are cell types. Okay? And cell types are usually, people think of cell types when thinking of division of function and how multicellular organisms are organized. Let me say also that, of course, this is perfectly relevant for the protist world. I mean, protists do have cell types. They have temporal cell types. Here we are talking about special cell types with some cellular memory devices that are not there in the protist. But we can also think of cell types as evolutionary units, in the sense that they are genetically individualized entities with discrete evolutionary histories. And questions about cell type evolution are really old in the literature. Like from the invention of the first microscopes, embryologists, zoologists, protistologists have been describing cell types morphologically. And they notice some striking differences, for example, between neurons in vertebrates and neurons in cnidarians or even in ctenophores. And this, of course, opened the possibility that some of these cell types are really old, that some cell types may even predate the divergence of major animal phyla, and therefore are hundreds of millions of years old. So the first major question in cell type evolution is, of course, about origins. When did cell types originate? Which is, of course, in essence, a question of homology. How we can discriminate between these neurons being homologous, and therefore similar by common descent, versus how easy it is to evolve these things convergently. And let me say that to this day, we are still scratching our heads and trying to figure out if these neurons here in ctenophores are homologous or not. And we work a lot on this. I will not be talking about this. But of course, to understand this macroevolutionary question, we need to understand how novel cell types, how cell type novelties, how cell types diverge, and how innovation occurs here. And we know that this is a major, an important factor in explaining animal evolution and animal adaptation. Just an example of a cell type that is one of the defining synapomorphies of an entire animal phylum, the cnidaria. So, of course, these days we can approach not only morphologically, but we can also think of cell types from a genomics perspective. And in a bit of a simplified way, we can think that every cell in an organism contains quasi-identical genetic information, and this information is differentially accessed in every cell through mechanisms of gene regulation. Transcription factors, chromatin processes that eventually unfold these different cell type identity programs. So, in a way, we can think of perhaps in a reductionist way, we can think of a cell type at its core as a regulatory program composed of transcription factors binding to their cognate sites in regulatory elements and controlling when, where, and how much a gene will be activated in development, in cell type identity, and so on. So, of course, this will be different between cell types, and we can, these days, hope to study cell types comparatively by profiling different aspects of these identity networks. Most commonly, we measure expression of these components. So, but until now, we've been in a situation where, especially comparing, and we're really envious of the situation on the genome side, that we've witnessed this massive, wonderful expansion in high-quality chromosome scale genomes across the tree of life. In contrast, our understanding of how these genomes function, what are the mechanisms involved in the regulation of these genomes, what is the diversity of cell types that they encode, and what is the logic of this encoding. How do you encode 10 cell types or 200 cell types? How do you encode stable cell types versus temporal cell types? It's much more limited, okay? But this situation has changed or has the potential to change thanks to the advent of single cell and other functional genomic technologies. What this allows us is basically with organisms that we can directly even take from the environment, we can measure different molecular activities. We can tear them down into their basic units, single cells, and measure different genomic activities, most typically expression. So what you see here, for example, is an atlas for a ctenophore. And you see that there is expression of variable genes, and there is this very strong, in these post-embryonic adult stages, these very strong expression signatures that identify these cell types. But we can also measure other activities. I will show you at the end. We can also measure the activity or the accessibility of individual regulatory elements genome-wide, and what is the relationship between these and the activation state of these genes. So all of this is great, and the potential is clear. And people usually show this kind of projection on how single-cell methodologies, in this case the single-cell transcriptomics, have expanded almost exponentially over the years. How many more data sets, cells per data set, etc. I'd always like to show that if we remove vertebrates, only vertebrates, from this picture, situation is less optimistic. Yeah? And there are a number of technical reasons on why. It's not like people have been trying, but there are a couple of technical bottlenecks that have prevented this. Of course, we can project these points as we should in a phylogeny. We did this recently. So this is a consensus phylogeny of the major eukaryotic lineages, number of species and genomes from goat. And here we projected where there are whole organism atlases in red, or some single cell data on specific stages or tissues. Okay. Of course, this is very much biased here towards animals, but even within animals, here you have your usual suspects. There are strong asymmetries and huge gaps in embryonic, larval, or whole adult atlases. And that's the reason why 2 years ago, with some people, also here from Sanger, we started discussing the possibility of something we call the Biodiversity Cell Atlas. So discussing whether this is possible, and if so, how we're going to do this, how are we going to decide which species, and what are the criteria for this, how we can accelerate a little bit the major bottleneck, which is the sample prep. So can we come up with a methods decision tree, and supported by validated protocols and unified QC metrics, how we can analyze and share these atlases in a way that is immediately accessible to everybody. And so for this, what we call phase 0, we got general support from the Moore Foundation. And of course, once we solve some of these issues, the fun part hopefully will come, which is to really scale up phylogenetic coverage in a systematic way. Okay, so that's the last thing I'm going to say about the Biodiversity Cell Atlas. I'm going to use this tree to highlight the group of organisms that we are interested in the lab, which is this part of the animal tree of life, the base of the tree. So what I hope I'm going to have time to tell you about today is 3 different stories on the 3 main areas that we're interested in the lab. The first one is about the evolution of genome regulatory mechanisms themselves. I'm going to show you a story on the origin of animal 3D genome. The second is more on cell type evolution, and perhaps an orthogonal take to the question of cell type homology, which is studying the evolution of specific cell type gene expression programs, in this case the neuronal gene expression program. And if I have time, I will show you, and I will try to motivate why I think it's important to not only look at transcriptomes, but also try to think of cell types from the regulatory identity embedded in the genomes, and our first attempt at doing this. Okay, so first a bit of the phylogenetic framework. As I mentioned, we work on non-bilaterian animals, essentially. We, of course, do comparative biology, so we always compare both with bilateria. And it's also important for us to look at the other side, so unicellular relative of animals. So ctenophores, sponges, placozoans, I'm going to tell you more about later, and cnidarians as well. This is perhaps our closest thing we have to a model species in the lab, which is Nematostella bectensis. So, and this first story is a bit motivated by some work that I was involved during my PhD and many others, which was when people started sequencing the genomes of unicellular relative of animals and looking for reconstruct the story of multicellularity genes. kind of the conclusion after years of research was that, well, there was not such a thing as a massive burst of innovation in new genes. There are no really animal multicellularity genes. So of course, an immediate conclusion from this is that, well, perhaps it's not so much about novel genes, but about how do you put these genes to work together in space and time. So people started looking at different candidate processes or mechanisms that could be involved in this regulatory complexity. The first one probably was the hunt for distal enhancer elements. This is kind of the foundational work in this, in the Nematostella from the lab of Uli Technau in 2014, that kind of like following the ENCODE paradigm using histone modifications, but also some in vivo validations. They showed that there are distal enhancer elements in Nematostella lenses. So then this was shown also in sponges and in other organisms. But there are other elements, as I will discuss, that could be involved in this. The capacity of insulation, sequence-driven insulation elements that restrict the influence of these enhancers, heterochromatic states that kind of shut down parts of the genome for the cell. So this could be, again, this idea of a memory device for cell differentiation. Or perhaps this has nothing to do with any special chromatin mechanism, but it's more in the gene regulatory networks that define these identities and how you can encode memory, what some people call network memory, in these programs. So what we did here, and I assume most of you are familiar with Hi-C methods that basically allow us to measure things that are in close physical proximity despite being in a certain linear distance in the genome. So we are surprised to see things off diagonal when they are touching each other. So these are methods that are generally used to assemble genomes these days, but also with high resolution methods you can also study all kinds of phenomena. And that's the interesting thing with 3D chromatin mapping methods. They are very powerful because you can see many things in there. But it is also confusing because many things generate patterns in a 3D chromatin map. Okay? And for us, an obsession is to discriminate between things that are involved in gene regulation, generally sequence-driven. There is an architectural protein, a factor that will recognize a sequence and will mobilize a molecular process, for example, loop extrusion, versus things that are byproducts of other processes in the genome. Yeah? Transcription, compartmentalization to some extent. At the end, it's the local chromatin environment that defines the segregation of, for example, active and passive chromatin domains. So when looking at these maps, and that's why we combined, and that's what we did, basically high-resolution chromatin maps with micro-C, if possible, of specific cell types, sorted cell types, decorated with linear epigenomic marks, accessibility, expression, histone marks, and so on, to try to make sense of what we see here. So what we did, we sampled 7 species, the 4 ones I mentioned before, and 3 unicellular relative of animals. Something we did when we started this, which was a great idea, we had some support from the Catalan initiative for the biogenome, and we resequenced some of these genomes. Like Nemiopsis was already sequenced in 2008 by the Joint Genome Institute. But it was broken in thousands and thousands of small scaffolds, so, and actually 1/4 of the genome was missing, actually. So we resequenced all these genomes. And again, let me emphasize that these are small genomes, or I would say standard invertebrate genomes, 200, 300 megabases, which means that micro-C was really instrumental here to have sub-kilobase resolution and be able to see things touching each other at these more micro scales. Also something in terms of expectations, these guys, you probably heard about CTCF, the major architectural proteins in bilaterians, invertebrates at least. In Drosophila there are other proteins, and they are absent in non-bilaterians, but there are other architectural proteins, as you will see. So we did all kinds of analysis. I want to highlight the main finding perhaps here, which was the presence of thousands and thousands of loops, of point interactions between parts of the genome, in ctenophores, in placozoans, also in Nematostella, a bit less. Nothing in the sponge that we look at. I don't have time, but I'm happy to discuss why we think this is the case. There is some distance dependency on this. And of course, again, we want to know what is contacting in this, in these loops. What are these loops bringing together? And in most cases, as expected, we find biochemical signatures of enhancers falling into promoters. So let me zoom in into one specific example, perhaps the most interesting, which is the ctenophore. So here you see the kind of all the data that we aggregated to look at this. And we started asking, you see we have some interesting methylation patterns, histone marks, etc. So we started asking, okay, can we, what's going on here? Now, can we describe this? So first of all, we see that in many cases, there are multi-contact loops, so basically multiple regulatory elements falling into the same promoter. What happens at the borders of this is what I kind of showed you before. There are typical marks of enhancers and promoters. Interestingly, there is SMC1, this is chipstick from SMC1. So cohesin is there, even though we find no evidence of loop extrusion. And this is something that we are looking at next. What are the mechanisms by which these are formed? Is it loop extrusion, or is it basically proteins that stabilize low-frequency contacts? And the more interesting part here is that we wanted to hunt for the architectural proteins that could be involved in this. Of course, we had no candidates, there is no CTCF, but we did the following. So first of all, we observed there was a very strongly enriched motif in most of these loop sites. Second, we saw that when looking at the attack, we see the typical footprinting profile, protection from fragmentation on these motif sites, which means that somebody is sitting there. Somebody is sitting in this. The other thing we observe is that methylation is very scarce in these genomes, but the occurrences of motifs where we don't observe loops seem to be methylated. They have higher levels of methylation. So what we did next is we do, for other projects, we do lots of chromatin proteomics in the lab, and we reason, okay, C2H2 are prime candidates for architectural proteins. It's known that in Drosophila, some insect-specific architectural proteins are C2H2s. So we generated chromatin proteomics data, and we reasoned, well, usually C2H2s being cell-specific, cell-type-specific transcription factors are usually in the low abundance range of abundance. But if these loops are all over the genome, this should be relatively abundant. And actually, we got lucky, and the most abundant, this is a retrocopy of this one, the most abundant factors, that we name them 10-4 tethering element proteins, CTIP1, seem to be the ones. And then what we did to validate both that they are indeed the proteins that bind to this motif, and also that they are methyl sensitive, just like CTCF, we did dapstic experiments where basically allow us to measure the affinity of these proteins for certain sequences, and also using non-amplified versus amplified genomic DNA, that methylation prevents the binding of this element. So, and what is the evolutionary history of this protein? This is a 10-4 specific protein. So it's present in all 10-4s. So in a way, for us, it's reminiscent of some like CP190, BIF32 proteins that are arthropod or insect-specific being involved in architecture. Now, of course, this opens the question of, well, is this an homologous process or not? Well, maybe it's different architectural proteins that can mobilize similar processes in the same way that you can think of different transcription factors activating the same process, which is recruiting polymerase. So let me just summarize what is the scenario that we reconstruct from our analysis. Basically, a common ancestor of animals, holozoa animals, and the closest unicellular relatives, where we basically, what we think that we observe is basically passive byproducts of other processes. Transcription, heterochromatin were present, so chromatin compartments. At the origin of animals, what we propose is the emergence of mechanisms for tethering distal regulatory elements, and in a way, this allows you to allocate regulatory information far away from your promoter. And what we couldn't find, and this is an open question for the future, is whether one can map the additional evolution of insulator elements, so basically an additional layer that would limit the range of action of these regulatory elements. And we know, for example, in vertebrates that these can be highly dynamic, and for example, during development. Okay, so let me move to the second story. This one is on single-cell transcriptomics, and this involves placozoans. So let me tell you a few things about placozoans. Placozoans were a monotypic phylum for quite a few years. So they were discovered in the 19th century, then rediscovered by Carl Grell in the 70s in Eilat. And actually, a clonal culture from this isolate has been going around, and it's the one that we have in the lab, actually. And they've been considered for a long time the simplest free-living animals. 6-7 cell types morphologically. It's basically like a 1 millimeter pancake with 2 cell layers, a columnar epithelium, a flat epithelium, and some tetraploid fiber cells in the middle. But people started looking for them in recent years. Actually, they are not as rare as one could think, and actually there are many species that have been described over the years. We even found them in Barcelona, in the port, or in the aquarium. And that's how people capture them. You put some glass slides, submerge them in coral aquariums, in rocky shores, and most likely they will jump if there is a nice microbial mat where they can feed on. And that's what they do. They basically have mucociliary motility. They go around, they find a patch. This is Pyrenomonas, the algae that we feed them in the lab, and this, you see the algae lysing. So they basically form a small pocket, they release digestive enzymes, and then they absorb. So they do extracellular digestion. So that's how they grow in the lab. So in the lab, we know they have sexual reproduction, but in the lab, we don't see them reproducing sexually. Nobody has seen an embryo beyond 128 cells. So that's how they divide. Boom, you've seen these two. Also notice that they are extremely plastic in shape. When they're well-fed and happy, they will grow, etc. So that's interesting. But what really kind of resuscitated the interest in placozoans was when the genome was sequenced. People started manually annotating what looked like neuropeptides in the genome. And when people synthesized them, this work from Dirk Vashouwer's lab in Lausanne, they added, for example, this LF peptide. You see how there are collective cell behaviors that are controlled by this peptide, in this case flattening and swirling. You will see that this SIFG amide, causes them to shrink and detach. There's all kinds of interesting questions on neural evolution. These guys have no neurons whatsoever. So who produces these neuropeptides? What is the diversity? What are the behaviors that are controlled? And so on. So I think I'm going to skip this. We used to think that the genomes, they are very compact genomes with very few genes, and they were very simple in terms of regulation. They are not so simple. But the important thing is that they are extremely synthetic, super conserved, but they have relatively few genes for an animal, 12,000 genes, because they basically have just one ortholog of each of your favorite genes, but no crazy expansions. Okay? So that's what we did with the help of Bernd Shearwater, Harald Gruber, that are basically people that go out, culture new placozoans and sequence them. So Xavi and Sebas in the lab generated four atlases for these different species. I'm going to just jump into the general details. We found the cell types that we were expecting to find. When we compare them across species, we see that transcriptionally, this is just transcriptional similarity, they are very, 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 very similar across species. But as usual, I should say, when we look at generate single-cell atlases for the first time in a new phylum, we found surprises. And in this case, the surprise was this group of cells here that like if I wouldn't know this is a placozoan atlas, I would say, aha, here are the neurons of this cnidarian or whatever. Because you see this combinatorial expression, some pan genes, but also some specific expression and so on. These are actually cells producing peptides. So we call them peptidergic cells. We describe 14 of them that are highly conserved across the 4 species. They have also an interesting story on how they are produced in the animal. I will not have time to tell you about. So they are defined by a very specific transcription factor code. Each of them has a different homeobox, but there are also different combinations, some pan transcription factors. We know that Notch delta is involved in the differentiation of these cells from the lower epithelium. And what do they have in common? What is this pan peptidergic module? So they express many elements of the presynaptic scaffold. Okay. Not only that, they also express several key enzymes in the neuropeptide processing pathways. So things that chop the propeptides, but also enzymes that add post-translational modifications, pyroglutamination and terminal amidation, and so on, that provide stability to our peptides. Okay. So we did mass spectrometry, small peptide mass spectrometry, and we could find in 60% of the peptides that we identify evidence of these post-translational modifications. Okay? So not only that, but this gave us a full catalog of candidate, kind of don't like to call them neuropeptides or secreted peptides in placozoans. So we could map the expression of their propeptides, of course. And then the question is, can we play guessing? What are they signaling to? And I told you before that placozoans have very few genes and no expansions, with one exception, which is GPCRs. They have a crazy expansion of placozoan-specific GPCRs. This here is how they are expressed across across these peptidergic cells, but also they are, of course, specifically expressed in the other cells, the effector cells that do things, digestion, motility, and so on. So then what we did is we teamed up with a group of Luis Serrano at CRG, and what they did is they co-modeled peptide GPCR pairs, some 18,000 pairs using AlphaFold 2, and then they performed docking analysis, this is what they are specialists on, where basically we were trying to predict the fit between these neuropeptides and the GPCRs. And we found 30 reciprocal best pairs, where the peptide likes this GPCR more than any other, and the GPCR likes this peptide more than any other. So this is a very conservative set. But from this, we could perhaps hallucinate a potential signaling network, paracrine signaling network in placozoans, which for me, the remarkable thing is the idea that this could be already a two-level signaling network where some of these upstream peptidergic cells, some of them are surely the sensory cells, they can sense gravity, presence of predators, light, and so on, signal to other peptidergic cells that in turn act over the effectors causing those collective behaviors that you saw. This is very interesting because this idea of a pre-neural paracrine signaling network, which in fact uses many genes involved in neuronal identity later on, fits very well with the hypothesis of the chemical brain for the early evolution of animal nervous systems, which is, yes, this idea of collective cell behaviors controlled by paracrine. This doesn't mean that this is an ancestral thing or a living fossil in any way. Placozoans have been evolving on Earth for as long as we have, but it is reminiscent, or at least shows how this could actually function in practice in this group of animals. So then, of course, we wanted to do comparative biology. So we also compared these cell type expression profiles with atlases from our lab and others available at the time. So again, this is the perspective of the cell type similarity. So what I'm showing you here, it is basically transcriptional similarity, multi-species transcriptional similarity, and unsurprisingly, given all the genes that I've been showing, we find strong similarity between these peptidergic cells in the 4 placozoan species and neurons as a whole, as a broad cell type in this. Question is, okay, what do we do with this? So assuming this grouping, what we can do now is we can transpose the matrix and look at the expression of genes, which is much easier because here we have orthology connecting across species. We don't need to make assumptions on cell type homology. And basically we can do ancestral state reconstruction on the gene co-expression. So here we are not looking at presence-absence of genes, well, we are doing in addition to, but all the fact that these genes are expressed together. And to cut a long story short, basically we reconstructed 4 ancestors with gains and losses using parsimony, and we found, or we mapped different functions, which I will summarize for you. So basically, what we infer is a common ancestor of placozoans, cnidarians, and bilateria, which is called parahoxozoa, that had in place a co-expression program with GPCR signaling, presynaptic scaffold, neuropeptide processing enzymes, and regulated vesicle release. So they don't have tons of ion channels, but they have the right ion channel, calcium channel, that controls the release of the synaptic vesicles. And the main innovation in neurons in terms of gain of gene expression, again, many of these genes are really old. Many are found even in guanoflagellates. But the co-expression element here is electric signals. So ion channels, we see tons of ion channels expressed in neurons, in cnidarians, in bilaterians, nothing here. A specialized postsynaptic scaffold, so specialized interface. So instead of paracrine signaling, the signaling is much more focused to this synaptic space, and also cytoskeletal proteins involved in neuronal projections. And this probably solves some of the constraints, of course, of these paracrine networks don't scale well when the animal becomes bigger, longer, etc. Okay, so this is it for the placozoans, and the last thing I want to talk about, and a bit motivate why going beyond transcriptional comparison. So I've shown you these macroevolutionary cell type similarity comparisons between sponges, placozoans, cnidarians, and so on. But we know we are doing many things wrong when we do these comparisons. And this is kind of a classic problem in Evo-Devo. We know these comparisons, these transcriptome comparisons, are dominated mostly by effector genes. And we know intuitively that many of these effector genes will be prone to convergent usage. Any ciliated cell will recruit the ciliary apparatus, but also divergence. And we don't have really good background expectations for how surprised we should be that two things are similar or not. This is because we don't apply. It's not that they don't exist. But given the samplings we have currently, we don't apply explicit evolutionary models for expression characters. The other perhaps even more obvious problem is that genes are not independent characters. Every gene votes independently when we do these comparisons. But again, think of the ciliary apparatus. It's a huge, ancient paneukaryotic machinery that all cells, ciliated cells, in absence of anything else, will resemble each other. And we see this in the data. We say, oh, of course, this is spurious, but this is methodologically problematic. So one potential solution to this is focus on regulatory characters as a better proxy for homology. This is the classic do transcription factor in situs between species to try to approximate homology. But then what do we really mean and what we can actually hope to measure, again, with this unbiased, many species if possible, etc. mindset. So I want to show you a last story, and this is, well, we've been working for quite some years on this. In trying to decode, and here there is no comparative element, it's just one species, but it's kind of important first-time work for us. And this is for our favorite cnidarian, Nematostella. Some facts on cnidarians, why people have studied them, and they are interesting. They are considered diploblastic, even though some people challenge this conception. So the origin of mesoderm, etc., have been studied because of that. They have neurons. We mapped back in 2018 around 30, 35 neuronal types, but no centralized nervous systems. They're generally slow evolving. People like to use some cnidarians, like Nematostella, for comparative genomics, because no matter what you measure, synteny, intronic positions, etc., they are relatively slow evolving. And this is, in our case, for Nematostella, most of them have indirect development with dispersive larvae. So we mostly work on adults. Now we're starting to work a bit on development as well. And these guys, we can culture them in the lab. They are quite easy. This is a system established already 2 or 3 decades ago by Mark Martindale and Uli Techno. And lucky for us, we were working again with a relatively suboptimal genome, and we were so happy, pleasantly surprised when actually Tree of Life released this chromosome scale assembly of Nematostella. So again, I think life would have been much, much harder without this genome. So we saw this, and we moved all our old data sets to the chromosome scale. So what we did here, I have some time. So basically we did single cell chromatin accessibility. And it was a project led by Ana Maria, PhD student, and Marta, postdoc in the lab. So where basically what we use is single cell fragmentation, and basically the game here, these are extremely sparse data sets, so the game is to aggregate them quickly, cluster them quickly, and then work on pseudo-bulk tracks, as illustrated here. So in practice, what we did is we sampled over 65,000 single cell ataxic profiles. We reduced them grouped them into metacells. This is a device that we like to use in the lab for some interesting properties. And then came the most difficult part, perhaps, which is annotating them. And basically, so to provide some naming and colors into these clusters. The intuition here is the following. If we aggregate the accessibility of all the regulatory elements associated to a gene, this is generally correlated with expression of the gene. At least this is the case. We quantify this for 80–90% of the genes in the genome. Of course, you need to also play the game of finding the right gene for the regulatory element and so on. But in these genomes, again, having a good genome, a good annotation, these are relatively compact genomes, this was not too difficult, and we could tentatively annotate some of this. Now, if we were to start this project today, we would instead use multi-omic profiling, so single cell transcriptome and accessibility from the same cells. So then you kind of move the problem of clustering to the RNA modality, and you just do the pseudo-bulking on the ataxic. But in any case, we get this beautiful, I mean, this is amazing to have the possibility to look across the whole genome and see regulatory elements firing up in different cell types. We also have gastrula stages here. We see all kinds of interesting configurations in terms of the regulatory logic of these genes. For example, a common modality in Nematostella is that 50% of the genes have constitutive promoters. So the expression is controlled by the action of additional elements. This is probably the gene with the most pleiotropic expression in Nematostella, GATA, and you see this complex, many elements, cell type specific, common, etc. And something, for example, that we saw, this is a GABA receptor that we could validate the existence of alternative promoters in Nematostella. One is driving expression in the fast retractor muscle, the other in neurons. Okay, so this is more on the regulatory logic, but then how we can distill this into different regulatory identity characters that then in the future we may hope to use for cross-species comparison. So one very straightforward thing to do is to measure transcription factor activity. So you would say, well, anyway, but you have expression, no? What is the added value here? The thing is that I always make the analogy that it's like if you were able to measure an enzyme, the expression of an enzyme, and in the same cell, some product that tells you about the activity of this enzyme. In this case, what you look at is that the aggregated accessibility of the binding sites, of the binding motifs, of this transcription factor in this cell. So you can have all kinds of scenarios where a TF was expressed during development, is not there anymore, but it has activated some sites and they remain open. Or you can have situations where you have a TF that is expressed, many of the signaling relays act like that, but it's sequestered in the cytoplasm and it's going into the nucleus. It will need to be phosphorylated by somebody. You will see that the sites are not active in the genome of this cell. Okay? So we can have this picture that kind of makes your life easier in identifying important transcription factors for this cell identity. Okay, this is a relatively... Of course, there is one very important detail here, which is you need to know what is the binding site of this transcription factor, but that's a story for another day. The other thing we can do is we can skip the transcription factor motif assignment and learn these motifs from the data itself. So we can do classic motif analysis, K-mer-based analysis. This is, in this case, is using K-mers, or we can also do deep learning. And what we learn from this is not only motifs that are important, but also combinations of motifs. Sorry, I forgot to mention that everything I'm showing you is for one example, that of the nidoside. So these are the top 4 most common configurations in nidoside-specific enhancers. Okay, so you see actually all of them has SOX2 because this is actually the pioneer in the neurosecretory lineage that probably is needed to activate those sites, but additional elements. And finally, you can try to put these two things together and try to model gene regulatory networks, which perhaps is questionable that they are interesting as an end in themselves, but there are some interesting things that you can do with these devices. For example, you can break a cell-type transcriptome into regulons, so in a way try to solve this problem of the non-independence of genes as characters. Okay, you can learn some TFs have auto-regulation, etc., etc. So you can make predictions. Again, there are many different ways of doing this. Happy to discuss the methodological details. What we used here is a relatively naive approach that correlates the expression across metacells of a certain transcription factor with the accessibility of a site, of regulatory element, times or modified by how strong is the motif using position weight matrices, the motif of this transcription factor in this site. So from this, we build these edges between regulatory elements and TFs and genes and TFs. So what we can do with all of this? In this case, we don't have cross-species data sets yet, but we can explore cell-type similarities within Nematostella. And I want to give you one specific example, and it's something that we've been scratching our heads about for quite a while. So these are the major cell types that we define in Nematostella with this data set. These are just similarities based on gene accessibility, which is similar to gene expression. Okay? And I want you to focus on the yellow stuff here, these two things here. And these are the fast muscles, the muscles that the Nematostella uses to contract or to move the tentacles. And this has been beautifully described by the group of Uli Tengnau, that one of them, the mesentery, the body retractor muscles, come from the endomesoderm, as one would kind of expect, while the tentacle muscles derive from the neurectoderm. And the story goes that, well, this happened because a group of HLH transcription factors got duplicated. You duplicate the TF. Now you can regulate it in a different context and bring all the downstream effector gene network. The important point here is that they are almost undistinguishable in terms of the effector genes that are expressed or accessible there. Indeed, when we look at these transcription factors, these are the two ones in the endomesoderm, this is the one in the tentacle, they do show completely different accessibility and some motifs that are typical from the respective lineages. What was a bit more surprising was when we clustered this based on accessibility of individual sites. This guy went with its ontogenetic friends, and this guy with the neurectodermal. So again, the same. And same when we do this using sequence models, where we can then train these models and use them to score regulatory landscapes of other cell types. So from this we can derive similarities. And again, perhaps even cleaner, we have this muscle here with the neurothermal derivatives, this guy here deeply nested within this. So what is underlying these structures? Well, the thing is that, again, when we look at accessible genes, expressed genes, they are very, 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 very similar, but the regulatory elements of those genes are highly dissimilar. This is one example, okay? So this means that this story of the co-option of 1TF and so on is much more complicated than that. For a gene to be activated in these different contexts, probably needs to be able to see a series of regulatory states during development that are completely different than this gene when it's activated in the endomesoderm. And this story is written in the chromatin, is written in the regulatory elements that are accessible, and of course, perhaps less surprisingly, is also written in the sequence motifs, in the regulatory sequences that are there. So in a way, this is, we need to see how well this holds in other cases. So we can clearly discriminate a well-known case of developmental homoplasy, and also means that perhaps written in these regulatory identities we don't know what is written there in terms of the evolutionary history of the cell, but it's definitely something written in terms of the ontogenetic history of the cell, which is kind of interesting if we think that for 99.9% of the species, we will never see an embryo. So maybe this is an additional layer of information that we can use to explore these kind of relationships. So just to finish, and then I hope there will be some time for questions, of a bit of perspective of where I think the field, evolutionary single-cell genomics, will be moving in the next years. I hope I have convinced you that the tools are there to start systematically interrogating cell types, not only transcriptionally, but also at other levels of defining this identity. And in a way, if we can follow the track of the genome availability and sample more densely, again, perhaps that was not the most smart way to start comparing a sponge and a ctenophore, but more dense sampling where we try to model the divergence of these characters, we can first of all start to bridge the gap between what we know, and these are very well-defined frameworks on genome evolutionary processes, and link these changes to and how they impact in this specific phenotype, which is the cell type program. So think of mutations in cis, in trans, structural variation, and formal models for divergence in gene expression, recruitment of modules, binding of transcription factors, and so on. And this is in itself extremely interesting, probably worth the effort on itself. But going back to the question at the beginning, even if we want to solve macroevolutionary questions like the homology of neurons, this is essential. Having evolutionary models, having expectations for how things should diverge, how be similar, how it is the likeliness of convergence, is essential to hope to more faithfully reconstruct phylogenetic relationships between cell types. And with this, I will finish, thanking all the wonderful people in the lab, also the BCA team at CRG and also at Sanger, and thank you for your attention.
Thank you, Arnau. That was lovely. Do we have questions? There's one up at the back.
Great talk. So I'm curious, how do ctenophore neural networks and cell development fit into your model of neuronal cell types? Do we know much about that?
Yeah, I can tell you something we are working very actively on is on a developmental atlas of a ctenophore with Pavel Burkhardt in Bergen. To try to say, well, I mean, let's compare neurons. So let me put it explicitly. Let's contemplate the possibility that maybe the story is that maybe some neurons in ctenophores would be homologous and others don't. So far what we can do is, okay, we have the kind of terminal neuronal transcriptomes. They don't resemble neurons in cnidarians or bilaterians at all. Let's look at the neurogenesis. Are there elements in the neurogenesis? I haven't shown that, but in placosomes there are some very interesting early steps that look like neurogenesis. And the other would be to look at the regulatory landscapes of those neurons. I don't have an answer so far, but they look really bizarre. Yes. So, 3 possible scenarios, not completely convergent, some convergent, some homologous, and completely homologous. I don't think completely homologous will be the answer, something in between or really convergently above. They are really, really bizarre. Like there are few features in a neuron that are super clear in any atlas, tons of ion channels, GPCRs, and so on, and ctenophores, they don't have any of these. Yeah.
So thank you for this brilliant talk. I have a question. So much of your assumptions rests on the phylum level architecture of the animals. And my question, how much variation within phyla, different lifestyles, and how much variation within different cell types, which is enormous, and so is variation within the phyla, may come into play, right? You get what you look for, and if you compare sponges with ctenophores, you find differences. But what do you expect as you go deeper into biodiversity genomics?
Yes.
Will this hold up?
Absolutely. I mean, this is what we are doing now is like comparative genomics in 2008, right? You had one sponge genome, one ctenophore genome, we have one atlas here, one So, of course, things will be very, like, for example, for cnidarians, for which we have quite a few atlases, we've done lots of corals and so on, there are many identities that are very conserved, but also we see a lot of interesting things happening with the neurosecretory apparatus. Or with the sponges, it's crazy. I mean, it's also true that we define them, it's a phylum, but the divergence between calcareans and demosponges is so brutal that the things that we see, we can barely align any cell type. So yes, and that's why my last point is like, we need to, or at least what I would personally like to do next, is we need to start working on very closely related species and see how the signals, conservation signals, fade away to set up the expectations for this type of comparisons. Yeah.
Cool. Any online?
Thank
Molly?
you for
Nothing online.
Sorry, thank you.
great talk there. I was wondering how important you think long conserved regulatory elements are in the context of regulation, long non-coding RNAs, non-coding elements, because that's involved in why are they conserved for so long across all these taxa?
You mean non-coding RNAs?
Yes,
yeah,
non-coding elements.
We are completely blind to them. So we have no techniques, for example, to measure that would be super cool. I mean, microRNAs, etc. I'm sure some of them would be conserved, but this is one of the biases of what we study, what we can study, in a way. Yeah.
Thank you.
Question here from Julie.
Thanks. With regard to your neuropeptides and your proposal of how they might have evolved into this neuropeptide signaling, maybe into neurons, I'm wondering in kind of more complex animals, is there any evidence that this early type of neuropeptide signaling still occurs, so that it's not actually only in neurons, non-neuronal cells and GPCRs?
Oh, yes.
Actually, these mechanisms, are they apparent? I'm just not aware.
Oh, yeah. There are still enterocrine secretory cells. I mean, the pancreas itself, which has a peculiar history, but uses insulin, which, for example, is a neuropeptide in cnidarians. So even some of the specific proteins are still working the same, yes, in non-neuronal context, absolutely.
Can I ask another one? This is on the your non-methyl binding in the first part of the talk, the CT, what is it called? CTEP.
Ah, yes.
You're probably aware, so that looks like it's binding a CG, like a CG sequence, and you're probably aware that there's a protein called CFP1 that's widely in higher animals, that's part of a complex that puts on H3K4 trimethylation. I'm wondering, is there any relationship between, and that's also a zinc finger protein.
Yes.
Could that be related? And is there a complex like COMPASS in your early?
Well, there's definitely, in ctenophores you can never say definitely, but most likely the COMPASS complex should be there. I mean, the marks are there. We looked at, for example, another candidate. I know I'm not answering your question directly, but there is this BEN P protein that Dirk Schubeler described, which was like, that's it. I mean, it's just methyl sensitive, CG rich. ctenophores are the only animals that don't have this structural domain and so on. So it could be that other proteins are involved there. Maybe something we are thinking to do now is, with some advances in chromatin proteomics, is to do the reverse. So not what the protein likes to bind, but what likes to bind to a certain sequence, to do pull down chromatin assays, and then maybe that would be a more unbiased way. Of course, we would find components of the compass case, because there are many promoters involved in this, so we will bring down all the promoter-associated complexes and so on. But there are plenty of regions in the genome, actually most of them, that don't have H3K4 trimethyl that are involved in these loops. So,
Okay, thanks.
Yeah.
Other questions? I have one about the non-homology of the two muscle types, those fast muscle types. So can you speculate on how many cell types we're defining in adult animals, which are actually formed by the convergence of two quite independent ontological pathways?
No. In this case, we have a suspicion on a neural type that could be also endodermal. Then it's also a question of how much weight or how much surprised we are that things are not completely segregated by a germ layer. No? And that's something that this is a very indirect way of approaching. Of course, with lineage tracing and other methods, we could really get quantitative in measuring that, and I'm sure there will be surprises there. But at this stage, no. I mean, I know in Drosophila there are a few cases of developmental homoplasy described for neurons, but in a systematic way, no, I don't dare to give you a number. Perhaps this is a very extreme case, because we are looking at the derivative of completely two different germ layers. And this makes it very apparent, right? So we are very sure that this is what it is. Maybe there are other things as well that we are simply...
a similar phenomenon is seen in C. elegans, where you get
the question is
differentiated cell types.
Learning from the specific examples, how you can generalize and whether it's possible to discover them in a completely data-driven manner, right?
Well, there's a final question for me then. If we just gave you a sequence, could you predict the cell types?
That's what some people are trying to do. I'm not so sure. I would be a bit skeptical. Let's put it this way.
Skepticism is good. So thank you again, Arnau.
Thank you.